Hello, everyone. Let's talk about this interesting theory that Omicron may have been the result of the use of Molnupiravir. Now, I'm not saying that I think it happened, and I think it probably isn't the way that it happened, but it is an interesting theory, and I think it's a something worth discussing. All right, so here's why we think that might be an issue. This is from a paper which I will link, and this talks about the number of unique spike mutations in the different variants that we've been exposed to. So here's alpha, beta, gamma, delta. And so you see alpha had 4 unique mutations, beta had 6, gamma had 8, delta had 7 unique spike mutations, and Omicron has 26 unique spike mutations. So that is way more mutations than any previous variant. So in most previous variants, you can, well, at least let's talk about how variants are discovered. Variants are discovered because many countries have what's called genomic surveillance. So they will take a random sample of their positive tests, and then they will sequence the entire viral genome, and then run an analysis to see which variant is being detected in the PCR tests. The PCR tests that are used in clinical practice do not detect specific variants. They just detect if you have SARS-CoV-2 at all, and it won't tell you which variant you have. So you have to do genomic sequencing to figure out which variant you have. So most countries are running these genomic surveillance programs where they randomly test the variants, and so you will know which variant you're having. And then if you see some drift, you can see the genetic drift within the variants, and there's like these sub-variants are identified. And most of them don't become relevant from a public health perspective because they're not any better than the other variants, so they don't take over. But Delta most recently was the dominant variant. For a while it was alpha, then it was beta, then it went to delta. Delta being the current dominant variant. And there is kind of you can see the genetic drift of the virus to the various variants. But with Omicron, it has so many different mutations, it's hard to tell, like, how did it actually become the Omicron variant? It seems like it appeared out of nowhere. So there's a couple of theories as to why this variant that has so many unique mutations may have come about. So one theory is that it may have come from a country where they were not doing genomic surveillance. So a lot of countries that are resource poor, they don't have the money or the resources to do genomic surveillance. So it's possible that there was a country where the virus was running through the population and basically picking up mutations, and since they weren't doing genomic sequencing, we didn't know about it until it showed up in a country that was doing genomic sequencing. So it was picked up first in Botswana and South Africa. South Africa does very good genomic surveillance, and they picked it up. And so that's one possibility. Maybe it was in a poorer country that was not doing genomic surveillance, and it was it had been brewing there for a long time and just wasn't discovered until it showed up in a country that was doing genomic surveillance. Okay, theory number 2 is called reverse zoonosis. So what that means is, remember I had that video about COVID in deer. And it's kind of a tongue-in-cheek video, but basically we know that SARS-CoV-2 can infect animals, minks, cats, deer, rats probably. So lots of other animals can pick it up. It's possible that they picked up the virus from a human, and then it ran through an animal population for a while. And since we're not doing genomic surveillance of animals, it could have been mutating in an animal source and then jump back to humans. So that's the reverse zoonosis theory. The other theory, and I think this is the theory that is the most currently, most people think is the most plausible theory, and it's one where we have another video about where variants come from. I'll link that in a card above, is that there is rapid mutational activity in an immunocompromised host. And the reason that is that in an immunocompromised host, you don't clear the virus very well, so the virus is replicating for a long period of time within one host. And during that time, the more replication there is, the more chance of error and mutations there are. And then through that, you can have selection pressure within a single person, and you can have a lot of different mutant viruses form within one patient with a prolonged infectious course, usually in an immunocompromised patient. There's several papers talking about how that happens and where it's been seen, where they see genetic drift within one person over a prolonged infectious course. That was discussed in a previous video, which I will have linked above. And then the interesting theory that we're talking about in this video is the idea that Molnupiravir may have caused Omicron or other variants. And here's the reasoning behind that. So as you know, Molnupiravir, and I'll link the video where we talked about Molnupiravir before, it's actually hydroxycytidine, and it's basically a nucleotide base that shifts back and forth in two conformations, and when they're trying to replicate the viral genome, if this gets incorporated, it's misread as various different nucleotides. So it basically causes what they call fatal mutagenesis. So it causes so many mutations within the virus that the virus becomes a non-functional virus. However, the downside is you are causing more mutations, and you could theoretically cause enough mutations that it becomes a better virus and is not fatal. And in fact, if you look at the FDA EUA application for Molnupiravir, and this is the first time I looked at it, it was very interesting. On page 32, they talk about the mutations that were seen in the spike protein during their clinical trial. So here it is, analysis of spike treatment emergent amino acid changes. So what they show is amongst the people that got treated with Molnupiravir, 34% had spike protein changes, and 23% in the placebo arm. So, you know, there are spike protein changes even without any treatment. And that's how variants occur. You know, the thing is always getting mutated. However, there are specific mutations with that they saw in more than 2 participants. And so they're listed here. So it looks like Molnupiravir causes enough mutations, and there's some kind of selection pressure to select for mutations that have clinical relevance. Like you'll notice this E484K is a very common mutation seen in a lot of the previous variants. So they said 5 Molnupiravir treated participants and none of the placebo treated participants had treatment emergent amino acid substitutions in the region of the N-terminal amino acids positions 139 to 145 in the N-terminal domain. This is an exposed region that is believed to be under strong antibody selection pressure. So that means that this is a mutation, that this area gets mutated, and it is an area that binds antibodies. So let's look at Omicron. Yeah, so there is this receptor binding domain. I think it's not showing it up here. But if I remember correctly, this antibody binding regions were areas that were substituted in Omicron. Then 2 Molnupiravir treated patients had treatment emergent E484K mutations. So at the 484 position, it got changed from an E to a K, which is a receptor binding motif associated with neutralizing antibody escape and is present in several important SARS-CoV-2 variants. So if you look at the SARS-CoV-2 variants, a lot of them had this E484 substitution. But they're not unique, so they're not listed as the unique mutation. They're kind of shared amongst a lot of them. And then 2 Molnupiravir treated participants had treatment emergent mutations in position 681, which is adjacent to the furin cleavage site. And so that is thought to be an enhanced infectivity of the Delta virus. Anyways, in the second trial, they noticed even more mutations. So again, you see that there's 35% in molnupiravir and 19% in the placebo. And again, more mutations that are seen in multiple people. And so from their conclusion is this: collectively, these analyses indicate that molnupiravir treatment may increase the rate of emergence of SARS-CoV-2 populations with amino acid changes in the viral spike protein, consistent with its mutagenic mechanism of action. So the way it works is it causes mutations. So they know that, and it does increase the rate of spike protein changes. However, there are many uncertainties regarding these findings and their clinical and public health implications. At the individual level, they didn't find anything bad happening. But basically, it says, in summary, concerns about the potential for molnupiravir to enrich low-level variants with spike protein amino acid changes within an individual patient may be low. However, it remains unclear if the potential for molnupiravir associated change in the SARS-CoV spike protein represents a public health risk. So they're saying in the individual patient, they didn't notice any problems, but we don't know if it poses a public health risk, which means that we can cause variants that get released out into the public and cause problems like we're having with, potentially with Omicron. So that is a pretty interesting theory, and I don't know. First, there's a slight chance that molnupiravir can cause mutagenesis within a person. And then two, I mean, that's very, very low. But two, it definitely causes mutations within the virus. And do we really want to use a treatment method that can cause viral mutagenesis and may cause the emergence of more variants? Seems a little risky. Maybe we should hold off on that. There are other treatment regimens available other than molnupiravir, and I think maybe we should put a hold on this until we understand this a little bit better. So again, not saying that Omicron is caused by molnupiravir. I mean, definitely there's no data that says that one way or the other, but it is possible. And it is interesting to note that during the clinical trials of molnupiravir, they did notice multiple spike protein mutations within their small treatment group, which is, they were only treating like 100, 113 people. I mean, just imagine if you're rolling this out to millions of people, the amount of mutated spike protein sequences is impressive. So that is another possibility. So interesting. So anyways, understand the four different theories. One is having the mutation happen in a country that is not doing genetic surveillance. One is reverse zoonosis, so the mutations happen in an animal population where we're not doing genomic surveillance. And then arising in an immunocompromised host. That's currently the number one accepted theory. And then four, possibly the result of molnupiravir treatment causing increased mutations in the spike protein of the virus. All right, so tell me what you guys think in the comments, and thanks for watching.